In this video, we're going to review a little bit about DNA and chromosomes. Why? Because the most essential thing that we're going to need to do for cell division is to copy the genome faithfully and partition it evenly between the two daughter cells. Because of course the genome is carrying all of the information to make the proteins and non-coding RNAs necessary to maintain life. Let's look at a human genome as an example. Here's a diagram of the 46 chromosomes of humans. Humans are what we call diploid organisms. This means that they have two homologous copies of each chromosome, with the potential exception of the sex chromosomes. So humans have 22 pairs of chromosomes that are called autosomes. Autosome simply means a chromosome that's not a sex chromosome. And humans also have, most usually, a pair of sex chromosomes, either XX or XY. And like I said, these pairs, at least the autosomes, are homologous. So for example, these two copies of chromosome 1 are homologous chromosomes. This means that they have the same genes at the same location and the same structure of the chromosome. Let's also remember that chromosomes carry genes. We're going to look at chromosome 3 as an example. So chromosome 3 has a lot of genes, over 1,000 is our best prediction at this time. We'll just call out 3 of them. So here's one. This is the oxytocin receptor gene. It codes for a protein called oxytocin receptor. You may have heard about oxytocin. It's a hormone and neurotransmitter that has a role in social bonding, reproduction, and childbirth. And it binds to the protein encoded by this gene, the oxytocin receptor, in order to do these things. Here's another gene that is on chromosome 3. This gene is the gene that encodes CCR5, which is a receptor protein expressed on the outside of white blood cells. And here's a third gene called rhodopsin. Rhodopsin codes for a receptor protein that has a role in the visual system. Let's remember now that humans are diploid and are going to be carrying 2 copies of chromosome 3. All right, now here's an important point, which is that homologous chromosomes have the same genes, but these genes could come in different alleles. Remember that an allele is a variant of a gene. So alleles arise by mutation, but then they can be inherited generation after generation. And this is going to come in handy when we talk about genetics. Okay, so let's look at the alleles here. Oxytocin receptor is at the top, and it's got a couple of alleles, big O and little o. These can have a real effect on the organism. Below that, we have a big C and a little c. The big C is going to represent our wild type CCR5 allele. The little c is going to represent CCR5 delta 32. You may have heard of this. This is the mutation that confers resistance to HIV-1. And at the bottom, we have a big R and a little r, indicating 2 different alleles of the rhodopsin gene. Some of the alleles that originally came from mutations in the rhodopsin gene can cause retinal disorders like retinitis pigmentosa. All right, so we have a hypothetical person that has 1 copy of each of these alleles, so they're heterozygous at each of these loci. Before a cell can divide, it needs to copy its DNA. All right, so let's take a closer look at a duplicated chromosome, which is shown here. This chromosome has been duplicated. We have to do that so that we can give 1 copy to each of the daughter cells. Each of the copies is going to be called a sister chromatid, or simply a chromatid in this figure. And each is an identical molecule of DNA. These chromatids are held together at the centromere, which is shown here in the middle. We also have telomeres, which are the ends of the chromosomes in eukaryotes, which have linear chromosomes. And on either side of the centromere, we have an arm. We just refer to the region on either side as either the P arm or the Q arm. The P arm is the shorter arm and the Q arm is the longer arm. All right, so with that chromosome anatomy, let's go ahead and copy our chromosome pair. So here is our hypothetical chromosome 3 pair with all of the alleles. It's going to undergo DNA replication, and this is what the chromosomes will look like afterwards. Now, I just want to note really quickly that on the left-hand side, when these chromosomes are duplicated at this time during the cell cycle, which is an interphase, specifically S phase, they don't actually look like this. They're going to be spread out and decondensed in the chromatin configuration of DNA. They won't look condensed like this until mitosis, but it is much easier to visualize like this. It's nearly impossible to see if we have it all spread out in the chromatin configuration. When the cell undergoes mitosis, these two copies will be pulled apart towards opposite poles of the cell, and then the cell will split in half such that each has one copy. And now we have two daughter cells that are identical to the parent cell. And of course, the same process is going to be taking place with all 23 pairs of chromosomes if this is a human cell. All right, so this is what it looks like with our alleles added, just focusing on chromosome 3. Here's our two duplicated chromosome 3s. At the end of mitosis, we're going to have one copy in each cell. So let's keep this in mind as we learn about the phases of the cell cycle, and especially the phases of mitosis, because it can be easy to get lost in the details. But this is what really matters, copying the DNA and dividing it evenly between daughter cells. So make sure you keep the big picture in mind as we start to dive into the details of the cell cycle and mitosis.